Hello everyone, today we're diving into cells to talk about an important pathway about glucose. It's glycolysis. This is our glucose molecule, our 6-carbon glucose molecule 1, 2, 3, 4, 5, 6. Whenever glucose wants to enter the cell, it requires something to transport it into the cell. In glucose, we have glucose transporters. We call it GLUTs or sometimes GLUTs. There are two main important glucose transporter type 2 and glucose transporter type 4. Glucose transporter type 2 is located on kidney and liver cells, pancreas, and glucose type 4 is located on muscle and adipocyte or lipid cells. Glucose transporter type 2 is insulin dependent. It doesn't require insulin to be activated. But GLUT2 is insulin dependent. We need insulin to activate GLUT4. Okay, now glucose enters the cells using GLUT2 and GLUT4. In order to keep this glucose into the cell, we need something to attach to it, something like an anchor to keep this glucose into the cell, and also in order to activate it. So we have our glucose here, 1, 2, 3, 4, 5, 6 carbon molecules glucose here. We add a phosphate group into our glucose, and we call this molecule glucose 6-phosphate. Turning glucose into glucose 6-phosphate requires an enzyme. It's obvious. And the enzyme is hexokinase and glucokinase. But it is important to note that glucokinase is only available in liver. Totally, we have 4 types of hexokinase. Hexokinase type 1, type 2, type 3, and type 4. The fourth type is glucokinase that is located only in livers. Okay, now we have glucose 6-phosphate and we're gonna turn it into the next molecule. What is this molecule? Oh, I have to add. We add this phosphate group to glucose. Where this molecule comes from? We use one molecule ATP and use one of its phosphate groups to attach to glucose. So one ATP used during this step. Now, in the next molecule, we still have this one phosphate group on the 6th carbon. So everything is looking the same. But we have an enzyme here called phosphohexose isomerase. This molecule helps to isomerate glucose 6-phosphate. It means that the number of carbons, the number of hydrogens, the number of oxygen are the same, but the structure, the 3D structure of the molecule is changed a little. And we call this molecule fructose 6-phosphate. So glucose 6-phosphate turned into fructose 6-phosphate. In the next step, we have an important enzyme called phosphofructokinase type 1. Phosphofructokinase type 1 add another phosphate group to this molecule, and this new molecule called fructose 1,6-bis-phosphate. Fructose 1 and 6-bis-phosphate. And we add this another phosphate using another ATP and turning into ATP to attach another phosphate group to fructose. In the next step, our 6-molecule carbon is split into 3 carbon molecules, and they both have their own phosphate groups. This one is dihydroacetone phosphate, and this one is glycerol aldehyde 3-phosphate. I have to apologize first because my main language is not English, and I might be sometimes weird in pronouncing some molecules and enzyme. I'm really sorry for that. I'm trying my best to pronounce in the best possible way I can. Okay, we have dihydroacetone phosphate and glycerol aldehyde 3-phosphate. The enzyme that do this is aldolase. Aldolase split this 6-carbon molecule into 2 3-carbon molecules, and each one has their own phosphate group. And they also can turn to each other by an enzyme called triose phosphate isomerase, and this is another isomeration. Okay, in the next step, when we do glycolysis, dihydroacetone phosphate prefers to turn into GA3P. So we have 2 molecules of GA3P here. In the next step, the GA3P molecule, this 3-carbon molecule, turns into another molecule, and it has another phosphate group. Okay. This new molecule, we call it 1 and 3-bis phosphoglycerate. 1 and 3-bis phosphoglycerate. And the enzyme in this step is called glyceraldehyde 3-phosphate dehydrogenase. Remember, whenever we have a dehydrogenase enzyme, it means that an electron transporter, NAD positives or FAD positives, is using in the steps. In these particular steps, we are using NADA positive to get to rip off electrons from this molecule, and this NAD positive turns into NADH. And another phosphate adds to this molecule. But this phosphate group is not coming from ATP. If an inorganic phosphate group, a free inorganic phosphate groups attach to this molecule in order to create this new molecule. And as I said before, we have 2 molecules of GA3P. This is why I write 2 NADH, because 2 glyceraldehyde 3-phosphate requires 2 pi, and it uses 2 NAD positive to create 2 NADH. So we have 2 1,3-bis phosphoglycerate here. In the next step, what we do? In the next step, we create a molecule called 3-phosphoglycerate. What has changed? A phosphate group has gone. How an ADP molecule comes here, gets 1 phosphate group, and turns into ATP. In other words, we produce energy in this step. We create an ATP by attaching a phosphate group to ADP. And the enzyme is also important for us. We call this enzyme in this step phosphoglycerate kinase. In the next step, we have this 3-carbon molecule, but the phosphate group location has changed. Now it is located on the 2nd carbon. So let's call this molecule 2-phosphoglycerate. These 2 molecule phosphoglycerate requires an enzyme in this step, and the enzyme is phosphoglycerate mutase. In other words, we had a mutation here. A mutation changed the phosphate location from the 3rd carbon on 2nd carbon. In the next step, we have this molecule here yet, but something changed. This molecule called phosphoenol pyruvate, and we call this molecule PEP. The enzyme in this step is enolase. I don't know why I keep this word in Persian, it must be in English, but it is enolase. Enolase helps to turn 2-phosphoglycerate into phosphoenol pyruvate. In the next step, and in the final step, we use an ADP. We want to get rid of this phosphate group. In the next molecule, we do not have this phosphate group. An ADP helps us to get rid of this phosphate group, and we produce another ATP here. And the final molecule here is pyruvate, and the enzyme in this step is pyruvate kinase. So let's review. We insert a glucose into the cell and turns it into glucose 6-phosphate in order to keep it into the cell. We use 2 molecules of ATP here, and we produce 4 molecules of ATPs here. And at the end of glycolysis, we produce 2 molecules of pyruvate from 1 molecule of glucose. So it is important to know that 1 molecule of glucose can turn into 2 molecules of pyruvate. So this pyruvate can continue in different pathway and different destinies in one way. If we don't have any oxygen in our cells, in other words, if the cell be in an aerobic condition, an NADH is used here to send electrons to pyruvate molecule, and NADH turns into NAD positive, and we produce a molecule here called lactic acid. This step is heavily regulated by lactate dehydrogenase, a very important enzyme. But the other destiny of pyruvate is in aerobic condition. In aerobic condition, whenever the cell has oxygen, it can turn pyruvate into another molecule, another 2 molecule carbon that we call acetyl coenzyme A. Acetyl coenzyme A or acetyl CoA is a molecule that can be sent into aerobic pathway to produce energy for our body. So this is totally glycolysis, turning glucose into pyruvate. You can have this illustration if you want. Just text me on Telegram, and my ID is below in description, and I'll provide you this illustration for you for free. And feel free to ask any question if you have. This is my actually first video in English, so as I said before, I might seem weird when I pronounce some words and some mistake like this one can be happen. It's in Persian, but I try my best to improve my English pronunciation to talk more fluently. Thank you for attention, for your like and your for subscription. See you in your next video.